Hi, I'm Linda Richards, Chair of the Department of Neuroscience at Washington University in St. Louis. I'm also head of the Brain Development and Disorders Lab here in the Department of Neuroscience. And today I want to tell you a little bit about the work in my group. So we are interested in how is the brain formed and wired to function. So how is it formed during development? And how is that development sculpting the circuits of the brain? So we're interested in mechanisms in early brain development that set up the wiring of the brain and then how that wiring underpins cognitive function. We study the largest fiber tract in the brain called the corpus callosum. You can see the corpus callosum here in a person in a sagittal view MRI. And you can see it's a very large structure and it connects the two cerebral hemispheres together. So how does this structure form? What are the genes and molecules and cellular mechanisms involved? And then once it is formed, how does it underpin cognitive function? So people can be born without a corpus callosum, and this is known as corpus callosum dysgenesis. Shown here is somebody that has a midline disorder, and so the corpus callosum axons arrive at the midline, but they're unable to cross. And they form these large bundles of fibers on either side of the midline here. This condition occurs in about 1 in 4,000 births and is associated with around 300 different congenital syndromes where the person has a corpus callosum disorder and another problem could be in another organ system or another area of the brain. But some individuals have a corpus callosum malformation in isolation. That is, they only have the corpus callosum disorder. And these are the individuals we work most closely with in our cognitive experiments. So here you can see different people with sagittal T1 weighted MRI images. You can see the neurotypical corpus callosum here, complete agenesis of the corpus callosum here, and a person with partial agenesis of the corpus callosum. Here we're showing some tractography images from our MRI data, and the corpus callosum fibers are coloured in red here. Now in this person with complete agenesis of the corpus callosum, you can see the fibers now run from front to back because they were unable to cross the midline. Here, the corpus callosum is greatly reduced, and there are still these ectopic bundles of fibers running from front to back. So we're very interested in how do disorders of the corpus callosum occur and how do they affect brain function. So to really get at the underlying mechanisms, we work in animals, but we also work with people that have corpus callosum disorders. And we go all the way from the micro scale, trying to understand the genes and gene function at a cellular and molecular and developmental level. We also do calcium imaging of brain activity and pair this with trying to understand how the circuits are forming in the brain. And then we study this with MRI brain imaging in people and in animals and perform cognitive neuropsychological testing and animal behavior to see the outcome of these circuits. There are three main areas of the lab. The first area is to study activity-dependent mechanisms of brain wiring in a small mouse-sized marsupial called a fat-tailed dunnart. Now, dunnarts are an amazing animal to work with. They have their babies at a very underdeveloped stage, equivalent to around embryonic day 10 in a mouse or a 7-week gestational human, and their babies develop in the pouch, so we can study them while they're developing. We also have a number of animal models, mouse models, which allow us to understand the molecular mechanisms underlying brain wiring. but we're also beginning to do some behavior in both the Dunnart and the mouse. And this connects to our work in people that have corpus callosum disorders, where we're looking at the cognitive outcomes of this, as well as the genes that are underlying these developmental disorders. So here is my group, the Brain Development and Disorders Lab. If you'd like to learn more, please contact me. Here is my email address, and you can learn more about our lab from our website. And we'd love you to join us. Thank you very much for listening today.